So my presentation is on the nutritional and the physiochemical and microbial quality of cowpea mahewu fortified with cinnamon essential oil. So mahewu is a fermented maize meal drink that is non-alcoholic cereal beverage. It's consumed by a variety of South Africans, including infants. The total amount of mahewu consumed in South Africa ranges about 146 milliliters that's sold in South Africa. Cowpea. Cowpea is a legume that is rich in protein, vitamin A, and minerals. Cowpea is high in lysine amino acid, which the body cannot synthesize. And it's also required in the body for growth. And then there's cinnamon. Cinnamon is an essential oil that's high in antibacterial properties that are active against foodborne pathogens. So what we're basically trying to do, right, is to increase the protein content in mahewu. And this has been done in other forms of literature, like, for example, soy ogi, where sorghum was used to increase the protein content, and millets bambara, where bambara legumes were used to increase the protein content. So therefore, this indicates to us that it is possible in a fermented cereal beverage to increase the protein content using legume. My aim. So the aim is to improve the nutritional and physical quality of mahewu, fortifying it with cowpea and cinnamon essential oil. The objective is to determine the physical, the chemical, the nutritional composition of cowpea mahewu when it's being fortified with essential.
Yeah.
Oh, okay. Okay. Yeah, the next slide. Okay, sorry for the interruption. So basically the aim, let me start over. So the aim is to improve the nutritional, the physical quality of mahewu by fortifying it with cowpea and cinnamon essential oil. The objective is to determine the physical, the chemical, and the nutritional composition of cowpea mahewu when it's being fortified with essential oil. The second objective is to determine the amino acid of cowpea mahewu, fortifying it with essential oil. And then the third objective is to determine the microbial stability of cowpea mahewu when you fortify it with essential oil for a period of 30 days. And the fourth objective is to determine the consumer acceptability of cowpea mahewu, fortifying it with essential oil. So the experiment design, so basically this table shows that there's going to be 9 variations, with the first one being the control, 100% mahewu. And then okay. So the experiment design shows that there's going to be 9 samples. The first one being the control, which will have obviously 100% maize meal. And the second one will be 100% maize meal plus 600 microliters per kg of cinnamon essential oil. And the third one will be 100% maize meal plus 800 microliters per kg of cinnamon oil. And then the second experiment would be 70% maize meal plus 30% cowpea, and then so forth, 600 microliters and 800 microliters of cinnamon essential oil. And then the third section, sorry, it'll be 85% maize meal plus 15% cowpea, and so forth, with 600 microliters of cinnamon essential oil and 800 microliters of essential oil. So the making of cowpea. Firstly, cowpea was grated, it was cleansed, it was soaked in water for 24 hours, and then after that it was dehulled. And then it was dehulled, used physically, and then it was placed on a tray and placed into the oven for 45 degrees for 24 hours. The next day it was roasted until it turned a brownish color, and then it was mulled into flour. Okay. So the making of cowpea mayo is 1 pot maize meal to 7 pot water. You boil it for 90 degrees for 15 minutes. While doing that, you occasionally stir, and then the porridge is left to cool for 40 degrees Celsius. And then wheat bran is added at 0.5 concentration to porridge. And then the mahewu is fermented for a period of 2 days at 37 degrees Celsius. And then the mahewu sample will be pasteurized at 63 degrees for 30 minutes to stop the fermentation process. And then it's cooled and used for analysis. So the test that's already been conducted, as you can see, pH measurement, total titratability acidity, color analysis, fat analysis, carbohydrates, crude protein, syneresis, and total solid. Okay, so this table is all the results that have been gathered through this process. So as you can see from the mahewu alone, the pH is really high, but as you add mahewu with cinnamon and mahewu with cowpea, notice that the pH starts to decrease, and yeah, the pH starts to decrease. And also we notice that as the pH decreases, the lactic acid and acetic acid starts to increase. And when the pH is when the pH is low and the lactic acid or citric acid is really high, it shows that there's antibacterial properties, and it also shows that that food can be stored for a long period of time. The moisture is really high for the first one, for mahewu alone, and also the moisture is high for the 600 microliters cinnamon. And what we notice is the fact that a high moisture content means that the food is not stored at, the storage is not long. So the longevity of the storage, the longevity of the ability of food for the food to store is low. Therefore, the quality of the food decreases. But we notice, however, in cowpea, mahewu, 85 degrees and 50, the variation, it has a very low amount of moisture content, this compared to the mahewu. And this, however, indicates that it does have a potential to have a high shelf life. Okay, so this is the color analysis. Mahewu alone on this table, it shows that it has a high. It's high in color, as in lighter in color. As it decreases, the second one, it shows that it has a darker color appearance, and also we also noticed that it was also high in yellowish color compared to, as it goes down, it reduces in complexion and becomes, Jesus. Okay. Okay, one of our objectives is to increase the protein content, and this nutritional analysis shows us that the protein content has, however, increased when cowpea mahewu, a legume, was added compared to the first sample, the control, compared to the control. So obviously you can see in protein, because that's our main focus, is that the protein was 20.55 in percentage. But as we added more cowpea, it increased, and that was one of our objectives. So, and it increased by like 9% or 9%. Okay, so what's left to do to complete this research project is amino acid analysis and microbial analysis, and also sensory analysis needs to be conducted. Yeah, thank you.
A round of applause again for the first presenter of the day. Thank you for the wonderful presentation. We have about 5 minutes to entertain any question. Any question for the presenter based on this presentation? All right, thank you. And please, the next presenter should get ready. So, Mrs. Betsy Ajibade, PhD candidate of the Biotech and Food Science Department, will be presenting on quality improvement of Amasi and Ryazhenka. Thank you.
A good morning to you also. I welcome us this morning. I'll be presenting on quality improvement of Amasi and Ryazhenka. So when we talk about fermented food, we all know that it's the new normal these days, and due to many scientific research that has been published and that are still going on fermented food, we realize that people now focus attention on fermented food, and a lot of people are interested. And even the industry, the food industry, is actually focusing on fermented foods. In the US and the EU countries, fermented food is gaining ground, and people are actually, consuming a lot of fermented food. Also, there is a paradigm shift now that people prefer to eat the fermented version of a food rather than the normal food. Like, you prefer to eat fermented vegetable rather than eating ordinary vegetable because of so many benefits that are attached to fermented food. Also, economically, there are projections that are favorable for fermented foods. So when you talk about fermented food, milk or dairy products is one of those things, of those food that you can actually look into the direction. And why do we have to look at dairy products? They've always been part of us. We have always been taking them. And we all want to drink milk. And a lot of time, because of their taste, their odor, the color, and so on and so forth, we need to preserve all this. And fermentation is one of the best way to do that. Also, because of health reasons, we drink milk based on the fact that it's highly nutritious. And when we drink fermented milk, we even get more benefits, which is probiotics. Probiotics is the new and the new normal. Everybody wants to take probiotics because it has been proved scientifically that it has beneficial effects. Probiotics are good bacteria that are involved in fermentation process. Also, because of economic reasons, the fermented milk market is gaining momentum and is gathering enough resources. So why do we have to drink fermented milk? And why do you have to produce your own fermented milk personally and drink them? A lot of time, we don't want to eat processed food anymore. People are running away from chemicals that are being put into processed food. So people prefer to produce their own food by themselves in their own houses. So fermentation is one of the best ways to process our food all by ourselves, and we'll be able to avoid chemicals used in processing and preservation of food. So another reason is because there are different arrays of microorganisms, beneficial microorganisms that are present in this traditional indigenous fermented food. And it has been proved scientifically that they can benefit our health. Also, they are easily prepared at home. You can actually start preparing after this workshop your own fermented milk, and you can get your own probiotics directly without exposing yourself to processed food. So when we talk about probiotics, what is it about them? They have so many advantages health-wise, and it's help our gut microorganism. It's help the microbes in our gut to function properly. And scientifically, it has been proved that the microbes in our guts or in our stomach that help, or digestive system that help in the digestion of our food, have a direct effect on our health. And consuming probiotic has been shown to have effective way of helping our gut digestion and all other gastrointestinal diseases. Wow, that's it. I'm supposed to be sharing this. So when we talk about probiotics helps in maintenance of the population of microbes that are present in our stomach or in our digestive system. And also it helps, it has so many properties like anti-cancer property, it's prevention of bowel diseases, cholesterol reduction, lactose metabolism, especially for people that are lactose intolerant. Also, it helps immune enhancement, and it also have therapeutic effects against pathogens and can actually help in ameliorating some of these pathogenic infections. Also, there are beneficial flavonoids and bioactives in this. Sorry for that break. So like I said earlier on, that this fermented milk also contains flavonoids and bioactive peptides that are very beneficial to our health. So when we talk about indigenous fermented milk, we have them from different regions, and most of them are based on wherever they are being sourced from or wherever they are being produced from. From Africa, we have different names that are being given to this fermented milk, and it's based on the traditional or the indigenous people that are consuming this particular product. For us in South Africa, our own fermented milk is called Amasi, and it's one of our cherished cuisine that we normally take every time. So from other parts of the world, we have Ryazhenka, we have kefe, kumis, dahi, name them. So many of these fermented milk were actually customized for a particular culture or indigenous people. And now they are all becoming popular, and people are started seeing the benefits of some of these fermented. And one of the reasons why they are becoming popular is because you can actually do them on your own or even produce them in your own kitchen and consume them all by yourself. So why is this fermented milk have these microbes? They are they are natural inhabitants of this product. And when you subject them to fermentation, they begin their work by feeding on the protein and on the carbohydrate that are present in milk, and they started producing some metabolites that are beneficial, that whenever we consume them, they are good. So some of these good bacteria, phylum that we found in fermented milk is Lactococcus gonostoc. Or fungi are also involved. So also is yeast, and some of the species are being projected. So we have the acidophilus bacteria, the thermophilus, the debruchis, the cremoris, and so on and so forth. The lactobacillus, they are the most popular of these probiotics. So when we talk about sources of this fermented milk, we have different sources, and mostly they are from animals, and some are domesticated and some are, they are not domestic animal. Like we have the cow, which is the most popular. You can make your Amasi or Ryazhenka or kefe using cow milk. Is the most popular milk, of course. And we also have sheep, we have camel, we have goats, horses, donkeys, also yak, that are wild animal. People get their milk and produce their fermented milk through some of these animals. Also buffalo. And one of the most important things that we should put in our mind is that some of this milk are very, very nutritious, and they contain some fatty acids that are very good for our nutrition. So when we talk about Amasi, why do we take Amasi in South Africa? We take Amasi because most of the time when people get their milk from the domestic animal, they tend to, they don't last long. So people subject their raw milk to fermentation. And this fermentation are spontaneous. When I say spontaneous, it means that there is no special microorganisms that have been introduced into the raw milk. They were just being left, and the fermentation just occur naturally. And you always have arrays of microbes in such type of milk. You have fungal there, you have yeast that are also part of this type of fermentation. And some of the reason why scientifically that has been proved is that some of the reason why we say Amasi is a very good food, is a very important nutrient drink in southern part of Africa, is because scientifically it has been proved that they have therapeutic effect. When you drink them, you get relaxed, and you have, your stomach, you easily remove waste from your body, and they are so beneficial to you when you drink them. They are easily also digested, especially for people that are lactose intolerant. So when you talk about production of fermented Amasi, traditionally people use plastics, they use calabash, like I have on the screen. Sorry, this screen is not moving at all. So we have the calabash, in which case this calabash is being used to ferment this milk. This milk is left in the calabash for like 2-3 days based on the season and weather that we have at that particular time. You can use like 2 days or 3 days for the fermentation. And at the end of it, you have your Amasi. You can remove the whey, or you can remove the top part, which a lot of people doesn't like. And if you like it, you can mix it together and, into a consistency that is more flowing. Some people like it thick and some people like it, thin one, by mixing the on this slide. It's not moving again. Yeah, I'm not sure. So for the production of Amasi, you can do it domestically too, using a plastic that is covered, that has a cover. When you leave the product covering it completely, you can increase the alcoholic content. But when you just put like a muslin cloth on it, then the alcoholic content will be seriously reduced. So depending on your own taste, you can actually produce this. Between 3 to 5 days, your Amasi should be ready. So when we talk about Ryazhenka is one of the milk, fermented milk that is becoming popular, especially in Europe. And it's also one of the traditional drink that have been, that have beneficial health effects. And it's originating from the Caucasus, region around Russia, Bulgaria, Georgia, Armenia. And these days, it's one of fermented drink that people are looking at, that people are investigating, researchers are looking into, because it has so many benefits. And one of the things that is unique about Ryazhenka is that you make the fermented milk from baked milk. What do I mean by baked milk? Baked milk means that you subject the milk, the raw milk, into oven at 90 degrees C for 8 hours. And there is a kind of, reaction that occur, Maillard reaction that occur, and the milk become brownish and caramelized in color. And it is this caramelized milk that you eventually fermented using sour cream or any other source of bacteria or probiotics to turn it to a fermented milk product. So traditionally, it is being produced domestically by people, and you can also produce this type of fermented milk in your own private house. So the major microorganisms that is involved in the fermentation of Ryazhenka are the Streptococcus and the Lactobacillus. So for the production of this Ryazhenka, you get your raw milk, you put it in the oven at 90 degrees C. One of the key things about this process is that you don't allow the milk to boil. The milk must not boil. It will just be eaten and evaporating, losing water. And during that process, you have a kind of caramelized milk. And this caramelization occurs through Maillard reaction, in which case, the casein protein reacted with the free sugars that are in there, that are in the milk, and they form a brown, caramelized substance. And it makes this milk very, very interesting. As you can see in this picture, the milk started with this particular raw milk, and after it has been put in the oven at 90 degrees centigrade for 8 hours, you have this caramelized milk, and it gives this beautiful color, and it's actually very sweet. It is after this that you now introduce some of the probiotics that you will prefer to use. Some people use traditionally sour cream is being used to ferment this brown baked milk, and it's actually very tasty and very sumptuous, so to say. So when we talk about quality improvement of these two particular fermented milk, we talk about how we can actually improve them. We can even do it personally, traditionally when you do it in your own house, or industrially, because some of these products need to be, market. We need to market them. We need to bring it to people that doesn't even know their benefits. So some of the method that's being used, in the literature and in the industry, we talk about freeze dry or spray dry. When you have your fermented milk in a dry form, you can actually, put it in anything that you want to eat. You can put it in your baked products. You can put it in your soup. You can put it in your salad to complement, or to remove the unhealthy, parts of your meal and put a healthy one. And also, like I said, that they are mostly source of probiotics, and we know the importance of probiotics. So when we use a spray dried or freeze dried fermented milk, we can actually introduce it to any food product that we want to do. Also, freeze dry and spray dried fermented milk have been used in the production of ice cream, production of pastry products, and so on and so forth, to have a healthy version of some of this food. Also, we have the frozen fermented milk. Just like I said, the frozen one, use them like your sorbets, you use them like your ice cream to complement your dessert when you are eating. Also, you have the fruit concentrate, the fruit pieces, and flavors. There are in the market now some flavored fermented Amasi, some with strawberry, some with orange, some with mixed fruits. Those are some of the quality improvements. Also, we have Amasi that are being the live culture. I saw one at Woolworths that you can actually get the live culture of this probiotic in them. It means that when you consume this fermented milk, you have access to probiotics that can actually help the microorganism in your digestive system. So these are some of the ways that some of this product is being improved. Also, we can have them in a concentrated form, whereby you can now, have so much in a small, in a small packet and use it for other production. So what are those factors that can affect the quality of these fermented products? The choice of your milk, the standardization of the milk, the additives that you had, the deaeration, whether there is so much hair or absence of hair, the homogenization, the heat treatment, the choice of culture, media that you use for that fermented milk. Also, for the industrial people, the plant design can affect, the quality of the fermented milk. Also, the microbial load is very, very important. Pathogen is one of the biggest issue of quality improvements. When we increase the quality and it's allow pathogens to be multiplied, there is no need to increase or to improve any quality. So microbial load of a milk before the starting of the fermentation is also very important. So these are my recommendations. You can make your own milk, just like I said. After this workshop, I want us all to try to produce our own Amasi with spontaneous method. And we should also note that we need to be seriously hygienic about it, because like we know, milk is very nutritious and it actually attracts pathogens. So when we want to try to do our own Amasi at home, or we are trying to, try our own thing, we should be very conscious of that. Fermented products is a way of the future. So please, you can invest, you can be an entrepreneur, to bring out some of these fermented products, because they are gaining momentum and people are actually recognizing them as a source of good and healthy food. Also, researchers should also still go into the research that will benefit all these fermented products. So also there are health benefits, which include gut microbiota improvements. Probiotics are still beneficial. We can still source some of these beneficial microorganisms from this fermented milk. These are my selected references. Thank you very much for listening.
Thank you for your presentation. We have a few minutes to entertain questions on the presentation. Yes.
Thank you for that question. One thing is that milk in the shop usually have their expiring date. And don't forget, there are different types of milk in the shop. You have the full cream, you have all other types and variety. I think what you should look at or look for is the raw milk. If you can get the raw milk from the shops, then you are certain that you, at the end of the day, you have good Amasi. Raw milk is different from homogenized milk or pasteurized milk. So if you can get the raw milk, at least you can, I believe you should get it at Woolworths, because I bought some at Woolworths. Thank you. For the first question, I would like to remind you that the lactobacillus, one of the main probiotics that are being used or that are involved in fermentation of milk, they have pathogenic properties. They have antimicrobial properties. You know, Lactobacillus lactis has even been researched to have antimicrobial property in Amasi. There's a research that have been published. So if I tell you that you should be hygienic when you are doing your traditional fermented milk, yes, you have to. But at the same time, the process is safe because it has been proved scientifically that some of the probiotics actually have antimicrobial properties, and they can actually protect your fermented milk, especially when you put, after production, and you put them at 4 degrees, between 7 and 4 degrees centigrade. Then for the second question, you are talking about freeze dry. That how do we, protect the, integrity of that fermented milk when we subject it to such process? Well, for freeze dried, we know that it's actually, it's a very good method of drying food product, because most of the time, all the substances in your food is even for research purposes, we are advised to freeze dry, because when you freeze dry, you still protect the integrity. Mostly, there are research work that have come out that you have 99% preservation of all your metabolites or whatever that is still in that particular sample. So it has been proved. So that is why we, advocate that if such is used for milk, of course, you can still get everything that's supposed to be in that milk. Am I able to answer your question? Yeah, for the even some of this, all of these fermented milk are based on the area whereby they are being produced. When during the winter period, you can use like 5 days to ferment. During summer, you can use like 3 days or 2 days. So based on the region, the relative humidity, the temperature, and so on and so forth, we affect the outcome of especially when you don't have any condition, if it's just normal room temperature condition, unless you set all the conditions, that is when you can get a constant fermentation during a period of time. It has been proved scientifically too, that it depends on the area and the environment and the conditions of your fermentation. Thank you. It must be controlled.
Try to find out the microorganisms that are responsible for indigenous and now use them in a controlled system. That method safe pasteurize them. Show the whole
Pathogens.
flora, all the things that are off table. That's what I was just trying to say. That's one of the things we need to tell like we need to try and help them to get those culture and let them know how they can improve.
Yeah, for the traditional method, they actually pasteurize their milk before leaving it, just like that. Even the method that I showed us, there's a place where you need to eat the milk. You eat the milk at 100 degrees centigrade for 5 minutes. So it's also still part, the eating is also still part of the process. So you don't just leave it like that. So thank you.
Question.
Um,
The question from those joining us online.
yes, from Pathology, he asked, how do we bring awareness of fermented foods to the rural dwellers who know nothing about microorganisms?
Thank you very much. Prof also still just mentioned it just now that even people that are in the rural area, they also need, orientation about the fact that this fermented food and milk need, to be pasteurized and homogenized, or at least eating it before they subject it to fermentation. I think one of the things that we can do is, sensitization. They need like a visit, like an orientation, like this type of workshop that we're doing now. It can be played in communities and in areas to create awareness that though you like your fermented milk, you like to do it like this, but because of science, we found this, we found that. So we can actually use this type of being recorded, to show people that are in rural area that they need to, there is a kind of improvement that they need to effect in their fermented milk. Also, there are companies in this country that are actually producing culture media for Amasi. I tell you, those companies that are producing the mass or Amasi that we saw in shops, they use starter culture. And when we were even doing research in the university, we buy from these companies. So they too can actually help. The companies that are producing these starter cultures can actually help those in the rural area by going there, because they are also going to make money when they started buying this culture media. Even we ourselves, starter culture, pardon me. So even those of us that want to produce our own traditional fermented milk in our own small kitchen, we can actually also buy this starter culture. Is a serious business and it's a good business because people are getting awareness about this probiotic. I don't want to go to the shelf and be buying dry probiotics. I would rather eat it in food. It comes with food and I consume it. Thank you.
Thank you once again for your presentation. We are still following the mandates for this workshop. Most of these fermented foods were actually invented by our forefathers without the current knowledge that we have. So it's our responsibility now to improve on them. It's our responsibility to explain the sciences behind all of these different fermented food that our forefathers have made for us. So we'll be having our tea break, but because of our time, we'll just allocate like 10 minutes for every one of us to grab their food. Then you can please come back to your seats so we can optimize our time. So basically, while you're having your tea or your snack, we'll be having presentations concurrently. So it's 10:40 now. By 10:50, our next presenter will be right here for the next presentation. So for now, we can go to the different feast tables to grab our breakfast and kindly come back to our seats. Thank you.
Recording in progress.
Assess it. How do we even assess it? Where's the flash?
Flash channel. Then flash it.
Flash and
It's the one from USB. Therefore, we did so. I just put it together. I'm not sure, but let's just go to
Video,
today's date.
video.
Today's date.
Okay, give it a second.
What you call it? Fermented something. Indigenous.
Okay, that's so we just click it.
Yes.
It's a slideshow. Look, I'll try.
Yeah.
Sorry.
Yes, sir.
But to share it on the, how do we share it on the, on Zoom? Because how do we share this on Zoom? Please Zoom. But we're trying to share screen. Which one? This is the one. Then we just hit play. Okay, maybe you not click play.
Yes, it's not playing.
The speaker. We are playing it.
Okay. Let me see. Oh,
Okay.
But it's not sharing. You are sharing.
Please, can you call the other guy, the technical guy? Yeah, the other guy to help us. This presentation has an audio, so it has to come from the laptop. So we want people to listen.
so now when you're playing that,
Yeah, it's not even playing. It's not slideshow. It's all going on.
Can you call it?
the sound difference. That's already telling you that those are signs of things that come having engaged.
Okay.
People have to travel about, have to do people, investments, have to
Microphone. Feel free to go to share another one. Don't play.
Good day, everyone. Good
Okay.
morning. Thank you for having me present this topic to you. And I want to thank the organizers of this workshop and webinar for their consistency in organizing this very important forum. Almost every year now, even for COVID for last year. So as to bring professionals and students together to talk about food microbiology, food safety, and other related disciplines. I'm so grateful to the organizers for inviting me. My presentation is on the microbial community of fermented production. Fermentation is a means of reducing or removing the limitations of soybean consumption. A lot of limitations have been adduced to soybeans and its consumption, the bean flavor, the some allergenic tendencies, and also the breaking down of some complex nutritional components. Fermentation has become traditional and is used to remove these limitations. So, good day, everyone. Good morning. Thank you for having me present this topic to you. And I want to thank the organizers of this workshop and webinar for their consistency in organizing this very important forum almost every year now, even for COVID for last year. So as to bring professionals and students together to talk about food microbiology, food safety, and other related disciplines. I'm so grateful to the organizers for inviting me. My presentation is on the microbial community of fermented soy-based products by high throughput sequencing. My name is Ajibola Bamikole, and I am with the Biotechnology and Food Technology Department, University of Johannesburg, South Africa. Introduction. Fermentation is a means of reducing or removing the limitations of soybean consumption. A lot of limitations have been adduced to soybeans and its consumption, the bean flavor, the some allergenic tendencies, and also the breaking down of some complex nutritional components. Fermentation has become traditional and is used to remove limitations of soybeans. That's why it contains high protein. So soy-based food products are gaining wide interests. They have always been consumed widely in the Asia, and now they are gaining wider interests in other parts of the world because of their health benefits, their benefits of their consumption. Both the fermented and non-fermented foods, soy-based foods, have high health benefits. And soy milk is water extract of soybeans, and it's made from grinding soybeans into slurry, and then sieving through muslin cloth into extract of soybeans, and then cooking to obtain soy milk. And fermentation brings about the improvement of its properties. Just like I've said, the fermentation is a means of removing limitations in soybean consumption. So fermentation of soy milk as an extension brings about the improvement of nutritional properties and reduction of different allergenic and adverse properties in it. So fermentation of soy milk can be through single or mixed microbial cultures of lactic acid bacteria. A lot of lactic acid bacteria have been used. Streptococcus, lactococcus, lactobacillus, bacterium. A lot of them have been used for the fermentation of soy milk, either singly or as mixed culture. And also spontaneous fermentation has been explored. They have been explored, but they have been sparsely documented. As a matter of fact, the traditional means of fermentation of soybeans may have been done in so many places, but the results have not been extensively documented. So, and soy milk has also been fermented with coffee grains. Coffee grains being a repository of microorganisms, both lactic acid bacteria, yeast, and other microorganisms which are involved in fermentation of foods. Soy milk has also been fermented with coffee grains, but their microbiology, the microbial fermentation, is still not clear. So, one of the ways to reduce the different attributes, the natural attributes of soybeans is sprouting. And sprouting before processing has been explored to reduce the adverse nutritional qualities. So that sprouting was also used in this experiment to also improve the qualities of soybeans and hence soy milk. Materials and methods. Sprouted fermented soy milk samples are prepared from sprouted soy bases. From our previous study, we have established 12 hours soaking and 5 hours germination as optimized conditions for germination. So we use these conditions to produce soybeans, and then we produce soy milk from it. And then from there, we subjected them to natural fermentation. 5 grams of sucrose was added to 100 ml of soy milk, and then it was subjected to fermentation. And then this fermentation proceeded at 6, 12, 24, and 48 hours, produced naturally fermented soy milk. And for kefir, soy milk kefir, we had 100 ml of soy milk, and then we added 5 grams of water kefir, and then we stopped fermentation at 24 hours. So it was 6, 12, and 24 hours. And then pure LAB and yeast colonies were isolated and counted using appropriate media. So for total genome DNA extraction, these methods described here were followed, and then the centrifugation methods were also followed. First, pellets were first observed, and then they were washed, centrifuged at 12,000 revolutions per minute to get supernatants. And then whole genome DNA was extracted using bit micro-EX gravity kit following manufacturer's instruction. And then the microbial community of fermented soybeans was determined using tag encoded NextSec and MISEC-based high-throughput sequence. NextSec for bacteria and MISEC for fungi sequencing. So the 16S rRNA and ATS regions were the ones amplified for bacteria and fungi respectively, while the PCR reactions were carried out in a short cycle PCR machine according to the method described by Creech et al. 2018. The PCR cycles used were described by Creech et al. 2018. And then on obtaining the DNA, amplified DNA, beg your pardon, library was constructed, and then Illumina sequencing was performed according to manufacturer's instruction, and then followed by processing, sequence processing, the processing of data obtained to understand the diversity of microorganisms and how they they occurred, yeast and lactic acid bacteria in the samples. Table 1 shows the result of the microbial counts of LAB and yeast from natural and chemical fermented soy milk samples. Like I've said, the samples were subjected to fermentation at 6 hours interval, 6, 12, 24, 48 hours. So that was the fermentation time that was used for this study. Samples were subjected to 6 hours, 12 hours, 24 and 48 hours. And from the results, we observed that the LAB count for sprouted products, that's those of naturally fermented soy milk and soy milk, were higher for all sampling intervals. And compared to the sprouted ones, we see that they are much higher. LAB dominated as the fermenting microorganisms in all naturally fermented soy milk and soy milk kefir samples. And they have significantly higher counts than yeast. And then we also found counts increased progressively in sprouted soy milk kefir, compared to others. It was observed that there were no yeast counts in sprouted soy milk kefir beyond the 6 hour fermentation. And this would be due to the production of lactic acid in high amounts. And the production of this lactic acid would bring about the accumulation of organic acids in the fermenting foods, which would have brought about the availability of dissociated acids for the neutralization of electrochemical proton gradients or cell membranes of yeast, and then causing them to die or causing them to be susceptible to death in the system. So that may explain the reason why we have no counts for yeast for sprouted soy milk kefir. Also, for kefir samples, fermentation did not progress beyond 24 hours. Because at 48 hours, we found that the food samples became very, very unacceptable in terms of sensory appeal. So fermentation was not allowed to progress beyond 24 hours. It also shows the bacterial diversity structure of fermented soy milk and soy milk kefir samples through by throughput sequencing. So we notice that sample F has the least diversity, while sample J has the highest diversity. Sample F is the spontaneously fermented sample 24 hours, and then sample J is the soy milk kefir fermented sample 12 hours. We noticed that famicutes became significantly expressed, and then Mucolostoc, Brucella, and Lactococcus genera exhibit increasing relative abundance depending on the type of fermentation that was going on. And also present in the whole community is the Bacillus family from phylum Famicutes. They are occurring spontaneously in natural and fermentation. And we also observe diversity varies among samples, but the predominant bacterial genera, the Famicutes and the Galactococcus, while the minor and trace population of bacteria that was observed include Propionibacterium, Bacteriobacterium, Bacteroides, Parabacteroides, Flavobacterium, and other genera that were present in the food samples. And it is observed that these genera are scarcely expressed in fermented soy-based foods. This figure represents the diversity structure of fermented soy-based foods. And 8 genera, which are Saccharomyces, Liposaccharomyces, Chlorospora, Pichia, Becerra, and Semiaspora, Mesconivera, with other minor ones were obtained. Fermentation by Saccharomyces, Liposaccharomyces, Pichia, and Becerra, as well as Chlorospora, was found to be more prominent across all the samples. And we observed that processed fermented soy milk at 48 hours exhibited up to 99.8% these microorganisms. It is also observed that while other microorganisms are expressed up to general level, Tolaspora quercum, Zygosaccharomyces parabili, and Decra animala were identified up to their species level. So A shows the simplified weighted unified principal coordinate analysis type of the samples in terms of microbial diversity, especially bacterial diversity. This figure shows the clustering of samples and how the bacteria, the fermenting bacteria, occurred, bringing about their together. These samples that are clustered to the right, are very close to themselves, to my right, which is going to be your right, that are close to themselves, show that they have a lot of fermenting microorganisms in common. And that is also shown in the figure, the previous figure, figure 1 that was depicted, that was shown earlier on. So as the stack shows how much of relationship that exists among them in terms of the fermenting organism. And we'll see that as we move to the left, the clustering reduces as we move towards the left in the graph. And also for the yeast clustering, what 2B shows the simplified weighted unified principal coordinates analysis by plot. But we can see here that the clustering is not so pronounced because they are more scattered. But then it is also obvious that the clustering to the right is more pronounced. And as we move forward, the clustering disappears. In fact, they are very separated from each other in terms of the occurrence of fermenting yeasts fermentation as revealed by high throughput sequencing. So in conclusion, that the use of high throughput sequencing helps to extensively show and characterize the community and diversity of fermenting bacteria, even the non-fermenting ones, because it is going to, since we amplify the 16S region, all the bacteria shown, and also since we have amplified the ITS region, all the fungi were shown. So help us to know their community in the natural and carefully fermented soy milk. And this is very important because we can identify some not too popular microorganisms that can be useful for fermentation, which can be removed, which can be purified and used for fermentation, controlled fermentation. The fermenting microorganisms obtained have not been widely employed in controlled fermentation of soy. So they can be made into starter cultures for controlled fermentation. That is the significance of this research, because we were able to discover potential fermenting microorganisms that could be used for the fermentation of soy milk and even other soy-based foods. Well, I want to acknowledge the Department of Biotechnology and Food Technology, Open University of Technology, and the Food Safety Research Group of the department, headed by Professor Oluwatosin Jabadini, for this opportunity and even the opportunity to carry out my PhD studies in that group and directly under supervision. I am immensely grateful. And I also want to appreciate the Department of Biotechnology and Food Technology, University of Johannesburg, where I currently work as a researcher and having exposure, required exposure. I'm so grateful for these opportunities. Thank you very much. These are some of the selected experiences from the work, and I am happy, I'll be very happy to receive comments, questions. Since I'm not physically there, I'll be happy to receive questions and comments and from participants of this workshop. And so I'll be very happy and looking forward to that opportunity. Thank you very much for listening. And once again, I want to thank the organizers of this workshop for inviting me and for allowing me to make this presentation. I have left my email addresses on this page for anyone who wishes to ask further questions or need clarifications. And you can also contact me through the organizers of this conference, of this workshop, and then we can take it up from there. Thank you so much.
That's it was a wonderful presentation by Dr. Ajibola. It's quite interesting that we both finished from the same high school at the same year in Ibadan, Nigeria. So this presentation has further highlighted what we have. That's further highlighted the gold mine we have with our various indigenous fermented food. That's like the previous speaker spoke about the production of starter cultures. So the study by Dr. Ajibola has identified microorganisms that's actually responsible for the fermentation process, which we can take further. We are here to show our contribution to knowledge. We are here to identify knowledge gaps. We are here to identify areas of further research, even areas of commercialization of our different research projects. So I would like to call on the next presenter, the person of Dr. Cyrus. Dr. Temitope Ekundayo, and his presentation is on bacteria and their role in indigenous fermentation. Thank you. Please, a round of applause for the speaker.
Good day, everyone. My name is Ekundayo Temitope.
Good day, Cyrus.
Okay. I will be presenting on the topic bacteria and their role in indigenous fermentation. This is just like a summary of what other people have presented, just for us to highlight some research opportunities that we can address. So I will not, the highlight of my presentation, outline of my presentation is as follows. Now, I will not assume that all of us understand or know what bacteria are. So I would like to define what bacteria are. Bacteria are unicellular organisms that we cannot see with our naked eyes. So in most cases, when we want to observe them, we culture them on media. and they produce colony. Those colony are multiple of millions of these bacteria. Thereby, we can see them physically. Just like Prof said the other time that we have the good one, we have the bad one. The bad one causes disease, deterioration of food, as well as food poisoning, a lot of disasters like that. But the good one is the one we exploit in production of antibiotics, as well as in production of food, as well as even in bioremediation of environments. So, and the most interesting thing about them is that they are common everywhere, on our body system, in the hair, virtually all the, wherever space or matter exists, you can find microorganisms, microorganisms as well as bacteria there. Now, based on their microscopic morphology, we usually characterize microorganisms into 5 groups. Some are spherical in nature. I mean, bacteria into several groups, like the spherical one, like in the ball shape, and the one in the rod shape, as well as the one in the spring or spiral shape. Some also appear like comma. So we have them, and aside from this simple structure, they can, the spherical one can join into making diplococci. They can form chains. The same thing with the rod bacteria. So the main characteristics of bacteria, why they are useful for indigenous fermentation processes, is because they have high surface area to volume ratio, which allow them to interface with the environment or surroundings, and thereby they can easily uptake nutrients from the environment, and whatever they produce from the system, they can also get rid of it easily. Then another advantage of this is also that they can easily reproduce via binary fission, divide and multiply in larger numbers. So another characteristic that make them useful is the mode of the nutrient transportation from their system. So they can, this is basically through concentration gradient, difference in simple division process. And when larger molecule needs to be moved into the system, they use active diffusion process. So another characteristics of them that make them useful in fermentation is that they are amenable to genetic manipulation, and also they can easily adapt to new environments. So, and they also can adapt to different environmental conditions and use different substrates, from complex to simple substrates. So they have this ability. Another thing is that they can produce new enzyme, or some of the enzymes are promiscuous in that they can act on different substrates at a time. So the fourth one, which is the metabolic diversity, they can obtain energy from different food sources, and they can also use different terminal electron acceptor. So that is why they are useful in indigenous food fermentation. So indigenous fermentation is just like one of our speakers already defined, that is spontaneous fermentation. You carry it out without introducing any microorganism. So, and indigenous fermented food are those ones we produce at home, the villages, and in small cottage industry. And the most interesting thing about indigenous food fermentation is that they don't require special equipment or special knowledge. It's just very few knowledge on how to process food that we needed to handle it. So, and indigenous fermentation is as diverse as human culture, tradition, and society. So we have a lot of them. Even within South Africa here, we have different culture, and the way we ferment food varies. So we have a lot of them, and just the start of emergence of microbiology give room for us to use the divine bacteria or microorganism to do fermentation, which we refer to as a control fermentation. But those indigenous knowledge is what we need to tap and use to get the new thing. So just like I said, that fermentable substrates are diverse and is very as human tradition. And in Africa here, we have a lot of variety of fermented foods. So this makes us having diversity that we can exploit for commercial purposes. So this is a map of Africa showing different fermented food from different aspects of it. So if we zoom up to this lower part, which is the Southern Africa, we can see coffee, cement, setim, and as well as sorghum beer, Amasi, and Togwa. A lot of them, we have it, just as we are very in culture and society. That is how we have a different fermented food variety all over Africa. The same thing with Asia, the Europe, and the Americans. They have their own indigenous fermented food product. So the question is, why indigenous food fermentation? The main reason for indigenous food fermentation at first is to preserve this food. For example, when you produce foods, they occur in there is a period of production, high production, that production exceeds the consumption or demand or supply in the market. So we need to preserve them. Another reason is to improve their sensory quality and nutritional quality. Then we improve their digestibility, and we also use it to detoxify some food products like cassava. They also contain functional effects, which are the probiotics. Then even when you ferment food, you increase their prebiotic property. Prebiotic property is the ability to stimulate gut microbiota. So these are the reasons we ferment or carry out any form of fermentation, and even at the indigenous level. So we also have, majorly we classify fermented food into two, the alcoholic and the non-alcoholic. And most of the one we consume, the fermented vegetables, soybean, fermented fish, milk, and so forth. Also, the alcoholic one can be from any food sample. We have honey fermented with honey or without honey. You can do this. Then if you want to make into alcoholic drink, then you do distillation. So Next, we will talk about the role of bacteria in fermented vegetables. So most of our speakers have talked about fermented milk and other food products. So now vegetables are edible leaves, bulbs, stems, and roots. And just like I said, they are prone to microbial deterioration. And the usual way to preserve them is to use pickling, sorting, drying, or fermentation. So, and vegetable fermentation involves, in vegetable fermentation, we are converting the starch and sugar within the vegetable into lactic acid. This is usually work of lactic acid producing bacteria. And just like other form of indigenous food fermentation, they just require low technology and low energy input, as well as texture or knowledge. So these are some of the bacteria that are involved in vegetable fermentation. We have a Lactobacillus acidophilus and Lactobacillus brevis, among others. So fermented vegetables, we can also have a fermented vegetable juice. And these are some of the important microorganisms that are involved in the fermentation, like Lactobacillus plantarum. In the case of garlic, cabbage juice, we have some Lactobacillus genera and some mixture of this. Then in the case of carrot juice, we can have a Lactobacillus plantarum and a mixture of Saccharomyces cerevisiae. So just like I said, there are a lot of these that can be fermented as pictorially depicted here. The fermenting medium is just a bottle covered with in which the vegetable is just kept and covered. So now spontaneous fermentation of vegetable indigenously can be carried out using dry sorting method, the use of brine. In the case of a dry sorting vegetable fermentation, this method is used for vegetables that have a high moisture content, and usually in a ratio of 190 kg of vegetable to 3 kg of salt is usually used. And an example of it is dry sorted pickled cucumber. In the case of brine sorted fermentation of vegetables, we use brine solution, which is 5 to 20% salt solution. And in non-salted fermentation, you ferment the vegetables without salt or brine. An example is fermented tea leaves. Also, the traditional way of preserving vegetables is peat fermentation. There you do it without addition of a salt, and this helps to keep the quality of the fermented vegetable. These are just a pictorial way whereby people do the fermentation. And example of bacteria that are involved is Bacillus subtilis, Propionibacteria species, and Lactobacillus that are effective in the
if you guys can join,
fermentation of
I want you to make me understand something.
What is vegetables?
Why is it that some of you can join and some do not have the ability to join? This is where the internet is not so good.
So we have
that the network provider is not so powerful. What exactly do you think?
Sorry, we are having an interference. Okay, now for
because I for example, there are some people who constantly come to us, but there are some that have not even seen them one day, and we've had 4 lectures. So, because I want to pick this up, I'll get to that.
Okay, the general process involved in vegetable fermentation is just you wash the fresh vegetable, like here we have cabbage as an example. Wash, you cut it, you slice it to different sizes of predefined grades. Then after shredding, you just leave it, keep it in the box, the container that you want to use for fermentation. Then the natural flora, the natural bacteria there will just act on it and produce the fermented stuffs. So we have an example of sunki, a non-salted fermented vegetable, and it's usually produced during winter or at low temperature. And the bacteria that are involved are Pemphidococcus pentosus, as well as Lactobacillus brevis. The same thing, Sinki as fermented vegetable. The bacteria involved are Lactobacillus fermentus, and usually the pH drop from 6.7 to 3.3 after the fermentation. So this, I will not talk much on this because people have, my previous presenters have talked on fermented milk and milk products. Those are the microorganisms, bacteria that are involved. The same thing here, I will skip this because people have talked on it, and that effect of fermented milk has also been handled. So the main challenge that we should have here now is, why are we organizing this symposium or research week? Is just to stimulate our interest into researching into indigenous fermentation process. And for us to do that, we majorly to create more awareness and the understanding of indigenous food fermentation and refine the processes, because when you don't handle it in the right manner, it can lead to contamination and as well as outbreak of infection. Then we need to disseminate the improvement, which we have documented, like the research Dr. Oyedeji presented. It shouldn't be left without getting it to the people that need the information. So we also need to document indigenous fermentation traditional knowledge. So to help us improve. So we also need to improve efficiency of indigenous fermentation processes, either by isolating and characterizing the associated microorganisms to produce starter culture. And we also need to investigate different pretreatment options to improve indigenous food fermentation, and also search for alternative options to improve indigenous food fermentation processes. And another thing is the process control, the selection of a microorganism just to predetermine the products right from beginning of the stuff. Then we can also do some culture manipulation and the cultural condition, optimize the condition to obtain good yield of the products. Then we can, just like we can develop a pure culture and using appropriate packaging. These are some of my selected references. I want to thank you for listening.
Thank you, Dr. Cyrus, for your insightful presentation. The presentation is open for questions and comments. Just a few minutes. Yes, Prof.
Can you hear me?
Yes, sir.
Okay, thanks very much for the helpful presentation. My question is, we note down the health benefit of these fermented foods, right?
Yes.
How do we pass the research and get them into the mainstream of business? Can I find some key or one of the other names? Amasi and Ogi in the supermarkets instead of buying instant oats milk, because I'm used to working when I was growing. Now I have to take oats.
Are you done, sir? Okay, thank you, Prof. And I think that is a very good question. Now, it's before we can get them into the market, we need to create much awareness on the health benefits and make it get to the right people that needs to get the knowledge. And if we want to commercialize it, that means we need to standardize our processes. So when we standardize it, then we can have good packaging and make it available in the market. So I think that is one of the ways we can. And for standardizing the process, we need to define this instead of using the common what is it called, spontaneous fermentation. For us to have good product, we may need to identify the essential microorganisms involved in such fermentation, develop them to starter culture, use them, optimize the production conditions. Because spontaneous fermentation, we just do it at room temperature, and room temperature can vary with the condition. For example, in the morning, it can be very cold. In the afternoon, can be very hot. And even in the night, sometimes it can be very hot. So the product integrity, if you are producing in mass, your product must have integrity, and that means we need to standardize the process. Thank you.
Yes, that is correct, right? But what if I tell you that this same response you just gave, I had it 20 years ago from very big professors with so many funding from European Union to even US. What are we doing wrong? Can I say something? Look at Amasi. It has been commercialized. How many brands of Amasi? How many brands of mahewu? South Africa has been able to show that it's possible. Other African countries also. But why can't we do the same thing? You know, why can't we have food in supermarkets? Why can't we have king? Why can't we have Nigeria? In England, they're also trying to, because we go to different Western, New York, you find Nigeria. So, but we need to still ask ourselves. Another thing I'm thinking about, when last did you buy food? Some of us, we don't appreciate what we have. We like Western products. So that's another problem. Young people, when they go value what we have to use it. Yeah. Prof is stop doing indigenous things like that. I'm a doing. People must value it. Like in Nigeria, where I grew up, they call it poor man's food. So, and if we value it and we produce it, the young people, the consumer will buy it. That's
So, Prof, I think we should as research institutions, we also need to look for industrial linkage. So I think that is the main problem we have. We do research, publish. Even I even had a case, some people will just go read publication and turn what people have published into a product without and get patent on it. So I think we research institutions, we are still lacking in the industrial linkage. That is the main disadvantage we're having.
And then to finish, just to be right, we are individual food society. We are passing that on. That are really coming to that. We will try our best before we exit. We are somehow halfway through the exit point. So we try our best, but I think something has to change. Definitely, either working even with policymakers. Sometimes it has to, it's not just a single element of research, policy, government priorities. We have to just give this rudimentary stage of research and characterization and have a vision of having a more viable product. And you mentioned something, the linkage with the industry, with the industry. So that's where the government comes in. You deliver, let's say, one comfort or a startup company is interested and such company receives a government support just to start it. So those are the things we need to have. I can bet you US won't come and support any product which does not grow in their territory. So that's the issue. Then we impose it on you in the process form. You have to buy it.
Then another thing is that one thing I've discovered is that government has some priorities in terms of health. Now, if we are doing any research into food, if we look at anything that, let's say, health related, in terms of alleviating this neurodegenerative disease, metabolic disorders like diabetes, such product, if we are able to formulate good product that have properties in that element, I think government will have no choice than to help. Thank you.
Any other questions? So with regards to the research done in universities and by scientists, how do we bring that information to my grandmother who is making a master effect, but does not know the help that she has been getting because she needs the children to get? How do I then bring the information? How to explain honestly the research to her in a way that she'll be able to understand? The barrier between research and something that is wise, and how do we as young scientists cover the gap?
Thank you so much. I think we should do more on science engagement. And science engagement is that we bring the research, the result of research into the community and explain to them in the common man language. So we need to start looking into that. And it's not without challenges because of the funding. When, let's say you want to go to one village in Durban, maybe suburb locations, you need some money, you need to, then for you, even you need people within the community that will be able to communicate with them if you can't speak their language. So we should dwell more on science engagement, bringing research to the people than publishing them alone. Thank you.
Please a round of applause again for our presenter. Science engagements, I think that is one of the things we are doing here. We are engaging ourselves. So in addition to what Prof and Dr. Cyrus has said, At the point where we are planning our research, we must make sure we are not just designing for research for publication purposes or just for graduation purposes. So at that point, we must design researches that will affect at least our immediate society. So that's very, very important. So we're moving on to the next presentation. It's by Dr. Titi Ajayi-Oba. She's just recorded a presentation and we'll just be playing it and listening. Yeah,
You want to remove this?
yes, sir.
Hello, friends. My name is Titi Ajayi-Oba. I welcome you all to the Indigenous Fermented Food Workshop and Webinar. Today, I'll be taking you through the topic titled Microbial Diversity and Potential Health Implication of Soil-Based Fermented Food and Beverage, OGI in Perspective. Indigenous foods are derived from plants and animal-based foods. They are native to specific location and consumed as part of traditional diets. Depending on location in Africa, the exposure and diet preference drives the transformation of food into diverse.
This one. These people are not here. Yeah. Let's try and get.
Hello, friends. My name is
It's a share.
Ajayi-Oba. I welcome you all to the. Indigenous Fermented Food Workshop and Webinar. Today, I'll be taking you through the topic titled Microbial Diversity and Potential Health Implication of cereal-based fermented food and beverage, OGI, in perspective. Indigenous foods are derived from plants and animal-based foods. They are native to specific location and consumed as part of traditional diets. Depending on location in Africa, the exposure and diet preference drives the transformation of food into diverse forms via fermentation. The indigenous knowledge was to reduce sugar content, increase shelf life, to introduce uniqueness, and improve storability. This practice has been passed over for generations now. Cereal play an important role in the diets of many consumers all over the world. Over the years, food products from different cereals have been modified via fermentation to increase food diversity. Various microorganisms readily metabolize carbohydrate-based substrates by way of fermentation. The three domains, which include archaea, bacteria, and eukarya, are usually involved. The community of microorganisms for any type of cereal fermentation largely depends on the substrate, prevailing environmental conditions, and the handlers. The type of cereal consumed depends on the practices in different geographical location, preference of consumers, accessibility to these cereals, as well as expectation in the final product. OGI can be made from maize, millet, and sorghum, or the combinations of the cereals as seen here. The combined cereal is traditionally believed to increase the nutritional value of the final product. A local processor is seen here processing OGI. This is the steeping period of the grains, which usually lasts between 24 to 72 hours. And this depends on the nature of the grain or experience of processor. Before we talk about microbial diversity, it is essential to highlight the factors that influences the microorganisms present in the OGI medium. Although fermentation is known to increase food diversity and nutritional value of the cereals, it is important to note that the initial state of these cereals matters and can influence the quality of the final product. Such factors include moisture content, the physical integrity of the cereals, growing practices, time and type of harvesting, post-harvest handling, storage management, and transportation practices. Apart from the initial quality of the cereals, the processing practices differ with location, and these influence its quality. For example, the steeping and fermentation of these cereals has significant impact on microbial diversity. Figure 3 shows the flow chart of preparing OGI, and the steps highlighted here show critical points that influence microbial diversity and subsequent interactions in the media. To start with steeping practices, cold water is used to steep the cereal for about 48 to 72 hours, depending on the level of dryness of the cereal. But the use of hot boiling water is becoming a practice, and this is done to hasten the softening process and reduce steeping time to about 24 to 36 hours. All these practices influence the microbial diversity and subsequent interaction in the cereal. Ogi is an indigenous fermented cereal slurry consumed by all age groups. The fermented cereal product yields an acidic supernatant, that is the stick liquor, but it's usually wasted. Sometimes this stick liquor is administered in the raw form for therapeutic purposes. The slurry is cooked before consumption. The type of microorganisms that will be involved in the fermentation of cereal to ogi are the prevailing microorganisms in the air, the microorganisms on the maize at the time of steeping, and the microorganisms present in the water used for soaking or steeping. Fermentation actually begins during the steeping period, and a lot of microbial interaction takes place. The duration of steeping is a critical factor. According to a report, the pH of the medium decreases. The fungal count decreases at the initial stage, but gradually rise after 48 hours, while the bacterial count increases and gradually drops after 36 hours. This is noted in medium where the stick liquor is not changed. There are natural and microbial toxins present in cereals. Natural toxins such as tannin and phytate, and microbial toxins, those are, example, the aflatoxins. These toxins are known to gradually leach into the steeping liquor during the steeping period. To have an improved quality of ogi, it is advised to change the steeping liquor every 24 hours to reduce the metabolic build-up from microbial interaction, as earlier said, because it may ultimately lead to off-odor of the final product if the steep liquor is not changed. A report showed that yellow ogi, that is the yellow maize, the yellow ogi from 48 hours steeping had the least residual toxin and best consumer preference. The blending process is also a factor, but it usually influences starch degradation. It's also important to note that during the blending process, temperature is introduced and it creates a suitable environment for microbial proliferation. The sieving procedure is very critical because the microbial community, the rate of microbial interaction, as well as the rate of souring, depends on the microbial quality of the water used for sieving, the level of hygiene of the handler, and prevailing temperature at the time of sieving. Recent reports have discouraged sieving because it reduces fiber content of the ogi products, and this step can also serve as a major route of microbial contamination. When looking at the microbial diversity, according to a report, 5 bacterial phyla, including Proteobacteria, Firmicutes, Actinobacteria, Bacteroidetes, and Lactomycetes were identified in the initial fermentation phase, but most dominant phyla that were present as fermentation progressed include the Proteobacteria and Firmicutes. For the phylum Firmicutes, the class Clostridia and class Bacilli were the most dominant and persisted in the fermentation media within 48 hours. In contrast, the phylum Proteobacteria, the gamma Proteobacteria were most dominant. Particularly, the family Enterobacteriaceae was observed to be predominantly in the media, and it persisted throughout the fermentation process. Apart from the class Clostridia, the class Bacilli, which of course is split into two groups, that is the Bacillus and Lactobacillus, have been reported. Notable geniuses include the Bacillus, Lactobacillus, Pediococcus, Neisseria, Lactococcus, as reported in another study. The Enterobacteriaceae identified from the gamma proteobacteria and some Bacillus species are useful indicators of hygiene and post-processing contamination. This shows that there are many microorganisms that co-ferment the ogi media. In another study, some yeast have been identified in the media after 48 hours of fermentation, and their presence continues to increase if fermentation is not halted. The diversity observed at the beginning and subsequent reduction during fermentation suggests release of some metabolites by dominating microorganisms. These metabolites suppress or destroy the growth of certain pathogenic microorganisms. However, some pathogenic bacteria are shown to be actively present even after 72 hours of fermentation. For the kingdom fungi, the phylum Ascomycota were reported to dominate the fermenting medium after 48 hours. Notable geniuses reported include Candida, Clavispora, Geotrichum, Pichia, and Saccharomyces. Candida crusei was the yeast most frequently isolated according to a report, while others that were found as fermentation increased are Clavispora and Saccharomyces cerevisiae. Some viruses have also been reported to be present at the beginning of fermentation, and this virus have persisted during the fermentation process. The sources of this virus may be from the air or the water used in processing the ogi. As indicated here, there are various species of bacteria and fungi that co-ferment the ogi. Some may be pathogenic with potential virulent factors, while some are not. Some of these microorganisms are also able to tolerate the metabolites from other competing microorganisms via various mechanisms. These potential pathogenic bacteria might have resulted from improper processing and handling, and this poses a threat to public health. The potential challenge here is that ogi medium contains both beneficial and non-beneficial microorganisms. The processing method employed is critical to the quality of ogi. The processing method actually determines the prevailing microorganisms. Ogi must be cooked properly to avoid or reduce the risk of potential food poisoning. Hygienic practices and standardized procedure are also very paramount, and the water source that used must also be from hygienic source. Research reports showed that regular decanting of liquor from ogi slurry after processing gradually reduces its quality because soluble vitamins and other nutrients are decanted alongside. It is advised to always drain ogi after processing and preserved in portions in the refrigerator. Small portions may be torn and used within 3 days. Although some potential threats have been identified, microbial behavior of yeast and lactic acid can result in the production of health-improving metabolites. Their synergetic effects also contribute to food functionality. Some yeast isolated during the fermentation of ogi secrete extracellular enzyme and also possess amylolytic activities, which aid the breakdown of carbohydrates in food to simple sugars for the use of lactic acid bacteria. The process of fermentation also aid in decreasing anti-nutrients in the food, beverage, and thus improve the bioavailability of essential dietary minerals in these food products. It has been noted that a wide range of microorganisms co-ferment the ogi media, and some of these microorganisms produce metabolites in form of antibiotics, bactericins, certain enzymes, polymer, and so on. Some of these microbial metabolites have potential application in health. Ogi have also been reported to have antioxidant properties, and their ameliorative effects have been tested in rats. Also, the steeping and fermentation of ogi is effective in eliminating majority of natural toxins, and reports have shown that the overall reduction of mycotoxins due to processing step for ogi is averaged at 90%. The production of polygamma glutamic acid producing bacillus species has been isolated from ogi liquor between 0 to 12 hours of fermentation. These bacillus isolates exhibit antibacterial and angiotensin converting enzyme inhibitory activities. The antibacterial activities of the polyglutamic acid were reported to be bacteriostatic, while the combined use of the polyglutamic acid from various bacillus species demonstrated bactericidal activity. Although the angiotensin converting enzyme inhibitory activities was low, ogi liquor represents a promising source for developing functional food with a beneficial impact on health. The importance lies in the benefits derived from the use of local resources. In conclusion, the consumption of ogi has various health benefits if processed under hygienic conditions and when they are properly cooked. The microorganisms and their metabolites can also have application in food and healthcare development. I want to appreciate everyone here. Thank you very much for listening. These are some of my selected references.
Thank you to Dr. Titi. I hope she's online hearing our applause. Maybe we should give her another round of applause. Thank you. So we have come to the end of our presentations, and I personally have gained a lot from these presentations. Have gained that our ancestors have actually done a lot. So now we have to develop on what they have done, because all of these products were invented years ago, probably centuries ago, without the current advancements in science and technology. And believe me, they were not discovered by accident. So we have to appreciate that effort. Then we have to build on it. And how do we build on it? We have to build on it by improving these products. We have to build on it by community engagement, science engagement. How do we better this product? How do we let people in the rurals know that with pure cultures or starter cultures, you can get a better product? So that is our job as scientists. I think we should leave like 10 minutes for engagement. Any questions, especially to be directed to our profs in the house? Yeah. Okay. Thank you. Okay. Please. Okay. Okay, Prof. Eric asked about the glycemic index of ogi, the very popular fermented food in West Africa. Judging from the glycemic index, which it says is above 90, is there anything currently being done, probably to modify the product to be more healthy, to be healthier for consumption, especially for people with diabetes and other related illnesses?
Prof, thank you for the question. Let me rephrase the question. Maize actually have a high glycemic index. Maize, flour that is made from maize, and maize now is being used to make ogi. So is it now good? Ogi itself, is it now good for people that are diabetic? So I think one thing we should understand is that the process of fermentation, the microorganism, the lactobacillus that are involved in the fermentation of ogi, they are actually, reducing the carbohydrates to lactic acid and so on and so forth. So when we look at the scientific results of what these bacteria are actually doing in ogi, we can actually say that ogi is a much better version of, as in that can be used by diabetic rather than maize flour itself. And also, I read in a research whereby some specific bacteria that can actually ferment carbohydrates to an end product, that is tolerable to diabetic or people that cannot digest or that want lower glycemic index food. So there are some specific bacteria that can actually be introduced into maize flour to make ogi or any other products that maize is being used to produce, especially during fermentation period. So some specific bacteria have been developed or have been discovered now that actually act on the carbohydrates that are present in some of these highly people that are as in products or food that contain high carbohydrates. Presently, I cannot actually mention any of them because I don't know them offhand, but then there are some of them that are being discovered now. Thank you.
Because whether it's enough, that level of activity of microbial is very enough to go on glycemic. Not work for something. Yeah,
Yes, thank you, Prof, for the question, and Dr. Betsy in waiting for the answer. Any other question, please? So I think probably the solution, one of the solutions will be production of starter cultures from those bacteria that Sister Betty just mentioned now. So that is one of the reasons we're here, and we have young and viral scientists here that can do that. And remember, at DUT, we are promoting innovation, entrepreneurship. We are trying to make sure that as a university community, we are contributing to the development of our region, our community. So it's very, very important that we take up these challenges. Please, any other questions? Okay. Please.
Mine is not a question, but comments on ogi. I think that fermentation reduces the glycemic index to some extent. And further fermentation, it's when you ferment ogi to some extent, it will having a kind of sour taste, which is one of the, if you compare it to sourdough used in the, that is usually recommended for diabetic patients. I think the maybe length of fermentation may reduce the glycemic index further. So I think is something we can research into. Thank you. Now, when you increase fermentation of ogi, you increase the length of fermentation, you have more of lactic acid, which turns it to more sour taste. So the sour taste is an indication that it might have reduced the glycemic index. You know, when the easiest, the layman way of testing glycemic index is the sugar taste or sugary taste. But when the fermentation increases and the product turns sour, then you are sure that the glycemic index is getting reduced. Thank you.
Dr. Cyrus, I think we have not answered your question, right? Yes.
Well, there is no report that when you ferment it further, you produce what is it, undesirable fermentation product. Personally, I have a personal, what is it called? We ferment it for long. Anyway, from the presentation, what we do is we change the fermentation water at some interval. So that might, from this presentation, I learned that leads to loss of nutrient. With that method too, you are reducing any pathogen, any metabolite that might increase as a result of continuous fermentation.
I hope we've been able to shed light on your, yes. So any other question? Yes, please come to the podium.
Okay, my question is that I've heard that the supernatant of the ogi, that is the water, fermentation water, that if you are stooling, you can take it and it's going to stop stooling. So I just want to ask, does anybody have any scientific basis for that? The supernatant of the ogi, when you drink it and you're stooling, maybe you have diarrhea, it stops the diarrhea. That's my question.
I think we have published work in that area, and it has been confirmed that basically is as a result of acidification of the gastrointestinal intestinal gut, the gut. So, and when acidification occur, then you are gaining more electrolyte compared to when you have when someone have diarrhea, is losing fluid. But when the acidic you take in ogi, the probiotics, some of the microorganisms, they have probiotics, and as a result of acidification of the gastrointestinal canal, you are also improving the homeostasis of the environment. Thank you.
Yeah, so building up on that, these are the methods that has been in practice for donkey years. Basically, in my own part of Nigeria, they use omyogi for anything, not just for stooling. For you have malaria, you take omyogi. You have typhoid, you take the water. So, and they actually work. So that's one of our essence being here. We have to provide scientific evidence. Why do these things work? You get it? So we have to provide scientific evidence. We have to research into these things and provide logical explanations. Please, any other question? Yes. Okay.
I have a contribution, please.
Okay.
Am I for please?
Yeah, we're hearing you.
Yes, I think a concept liquor refers to as omiyugi in Yoruba language. It is apart from being assisting body in regulating the body immune response. It also helps to maintain the intestinal microbial equilibrium. That is, when the level of a pathogen that might have invaded into the DIT through one means or the other, taking this omiyugi, it will help to stabilize the microbial, the beneficial microbial equilibrium in the intestine, thereby conferring GIT immunity. And also, like me, when I was in the village, when I take omiyugi, easily I see some heat, some kind of exothermic reaction coming out from my body. I urinate. I believe it's a sort of the therapeutic potential that present in this omiyugi. And naturally, we believe that omiyugi contain a lactobacillus species, which are been reported by Oyetayo Ifuta to be as a probiotics that enhance human immunity. I believe it is good if you can encourage our people to be taking omiyugi, having follow the proper quality control check while preparing the ogi. Thank you.
All right. And for the food technologists and scientists in the house, ogi is the same thing as mahewu, right? Oh, okay.
May I please make some comments?
Okay, please, we can hear you.
Okay, good day, everyone. Sorry, I'm in between lab sessions, but I'm happy to join. I just want to provide more background to the steep liquor benefits that metabolites that are also produced by the microorganisms during the fermentation of ogi also contribute to the beneficial potentials or powers this time of this steep liquor. So I know Dr. Titi has worked extensively on the characterization of the steep liquor. I don't know the extent of metabolomics that she was able to do while working on the steep liquor. So this is also an area of interest that we should look into. We should be able to characterize the metabolites that are involved, that are present. Metabolomics be targeted, it could be untargeted metabolomics to know, in addition to the acidic, the acid that is produced due to the activities of lactobacillus and other lactic acid bacteria, there are other metabolites that are of great benefits that can be looked into. So it's and of course other fermented products that have steep liquor that could be steeped. Their steep liquor could also be looked into, and their potentials for health benefits as possible. Thank you very much.
Yeah, thank you, Dr. Ajibola. And we have a comment from one of our virtual participants in the person of Omotade Ogunremi. Says, about the glycemic index of ogi, I'm of the opinion that the choice of cereals can play a role. Some cereals, e.g. millets, are known to be low glycemic index cereals. Also, choice of strains used to drive the fermentation has been found to influence the residual sugar content. Thank you, Omotade Ogunremi, for your comments. Please, more comments, questions? Okay.
Hello.
Yes, we can hear you, please. Go ahead.
Okay, thank you very much. So on the glycemic index, like I mentioned, the choice of cereal can play a role. So I also want to comment about the steep liquor, and I will come in from the angle of the probiotic potentials of such steep liquor. We should be aware of the fact that we are consuming live microorganisms with those steep liquor, especially lactic acid bacteria, and they are known to play the roles of producing antimicrobial metabolites. And beyond that, they could also go into the intestine and compete with these organisms that tend to cause diarrhea. They compete for space, points of attachment, and the likes. So they tend to limit their activities in the gastrointestinal tract. So the probiotic potential is an interesting angle. And I have a question. Maybe it could be taken as we go on with the discussion. Talking about the use of starter culture and this drive for fermented food to be used to populate the microbiome of the intestine, fermented food and microbiome of the guts. I'm just imagining and wondering, when we go for starter culture usage, we need not limit the ability of fermented food to promote this robust microbiome in the intestine. Just imagining if that could be addressed. Thank you.
That's a question for everyone. Anyone to answer the question? Okay. Okay. So the answer from Prof. Eric is the use of, he says it's a suggestion, the use of starter culture, standardization, and hygienic practices, probably a SOP if needed.
Thank you. I think the need for the use of starter culture is for us to guard against product integrity. Because when you do spontaneous fermentation, you can't get the same quality every time. So when you use starter culture now, you are sure of your end product. So before you can select starter culture for any process, you must have optimized, you must have start from the spontaneous fermentation first, characterize different microorganisms there, combine them, look at the one that can. Single strain might not be useful as a starter culture when you are dealing with fermentation. So I think there is nothing wrong with the use of starter culture, most especially when you want to commercialize your product. So the product integrity must be registered. What the consumer want to get must be documented. If you are using spontaneous fermentation, you are sure you won't have the same quality of product every time. Thank you.
It's always the same. Props, is it not true? We never even know about standardization. Every time we buy this food and we enjoy it. But that's why it has not been commercialized. We love it like that. No, we want to produce it.
Okay. So basically, moving forward, we have to standardize our processes. We have to make use of standard operating procedures that would document the components, the ingredients, the particular starter culture that will be used to ferment our various indigenous food. And we should keep it in mind that this workshop is not just about ogi, it's about all fermented foods. So the comments, the challenges, the opportunities that have been highlighted with regards to ogi also apply to your mahewu to your Amasi and Injera, etc. and etc. So at this point, I would like to thank all the participants for their patience, for their questions, for their comments. And I'd like to call on Prof. Tosin to give the concluding remarks. Thank you.
Well, we've come to the end of today's event. I want to appreciate everybody for staying to this point in time. We've heard a lot about indigenous fermented foods. I want us to support our own products. You know, I asked a question when I started the other time. How many of you have consumed fermented foods in the last 1 week? Maybe 2 people raised up their hands. And I think apart from consuming fermented food like yogurt that is common, we should begin to appreciate our own indigenous fermented food, like your Amasi, like your mahewu. Begin to buy it, begin to consume it. And we must also begin to encourage people to go into commercialization of these products. And we that are from other African countries must also learn from what has happened in South Africa, where they have industrialized, where they have commercialized it. We have a lot of indigenous fermented foods all over Africa, and we need to do something about it. I hope you gain 1 or 2 things, you know, from our event today. And I'm also want to thank everybody that has joined, especially people that join online, people that join virtually. Thanks for joining in. And also our presenters. Thanks for doing a good job, Leonie. You know, you did a good job. Betsy presented part of your PhD work. Dr. Ajibola, that presented part of the work that he did while he was doing his PhD at DUT, and currently is doing his postdoc at University of Joburg. Dr. Cyrus, thank you for that interesting presentation. And Dr. Ajayoba. Dr. Ajayoba was a postdoctoral fellow a few years ago, or even 2 years ago, but she's back in Nigeria. Thanks for presenting that, interesting presentation. And Deji, thank you for today. And everyone that has joined. Professor Eric, thanks for coming. And I think that's it. So we have lunch available downstairs. So we hope to see you some other time. We're also going to be organizing other, events before the end of this year. I'll let you know about it. So thanks once again. And Ibilala, thanks so much for what you also did. I almost forgot you. Thanks so much for what you did. Ibilala was one that did a beautiful flyer. And also Betty, they came up with the program. Thanks everyone for doing a good job. Thank you so much. Bye. Okay, I don't know whether she wants to say something. Okay, let us say, what do you want to say, Dr. Titi? Okay.
Dr. Titi, if you have any comments to add, please unmute your mic. Yeah. Okay, in the absence of any other comments, let's call it a day. Lunch, and thank you for your time.
Yeah. Okay, thank you.
Thank you. Okay, thank you.
Congrats.
Thanks so much. I'm looking for my key. Yeah.